Welcome back to MIP 302. Today we'll be talking about exercise 11, which is our immunology exercise. Specifically, we'll be talking about ELISA. So what does ELISA stand for? ELISA stands for Enzyme-Linked Immunosorbent Assay. And there are several different types that we're going to talk about, or that your pre-reading talks about. Direct ELISA, indirect ELISA, antigen capture ELISA, or antibody capture ELISA. Other useful terms to know here are antibody and antigen. So when we're talking about an antibody, or if I draw it, it's going to be depicted by this Y-shaped little image right here. But essentially, an antibody is a protein that's produced by your immune system, or your B cells specifically, right, to neutralize a pathogen by binding to antigens. Okay, so what is an antigen? An antigen is a toxin or a foreign substance which induces an immune response. Now this could be bacteria, a virus, a toxin that's produced by a bacteria or a virus. And I'm going to be drawing those, and they're going to be depicted by this blue little triangle here. So if you see one of those, that's an antigen. Alright, so what is a direct ELISA? So your book does a pretty good job in describing these, so I would really look and read those as well. There's also a nice series of images, I think on page 135. But essentially with a direct ELISA, we're trying to detect the presence of a particular antigen in a sample. We want to know, is that antigen in that sample or not? So if you wanted to measure something like the immune response to a particular antigen, what you would do, I've kind of drawn it out here. Here is your plate or your well, so whatever you're trying to bind to, maybe a tube. Here's your antigen that you're trying to detect, and then you would have a labeled antibody that would bind to that antigen. And when you would add a substrate into that solution, that labeled antibody would have an enzyme on it that would change that substrate from this form into this form. And that might be something that a machine could pick up, some type of machine reader, or you could see it with your eyes via a color change. In this case, it might be a red color change. So that's how that works. If you were working in a lab and you were thinking, okay, what are the benefits of doing this method or any of the other methods? Well, it really just depends on what question you're asking, on which one you'll do, and how much you know about your antigen and your antibodies that you're looking for. So one thing that you can think about for direct ELISAs, it's time saving in comparison to the other ELISAs because it doesn't have as many steps or components to it. And because of that, and because there isn't, for example, a secondary antibody here, you don't have any cross-reactivity with that second antibody. So that's good. But there's pretty low flexibility with this procedure. The primary antibody must be labeled. So if you cannot label your primary antibody for some reason or another, then this is not the method for you. Also, if the antigen that you're trying to look for some reason changes shape because of binding to your well, then this is also not something that you would be able to do in your experiment. Alright, let's talk about indirect ELISA. So this is the ELISA that your lab exercise goes into, like what you would actually have done in your lab. So it's probably the one that you need to spend the most time understanding. But essentially with an indirect ELISA, you're trying to detect the presence of a specific antibody directly against a particular antigen. So for example, let's say I'm working in the medical field, which some of you I know are aspiring to do, and I have a blood sample from someone and I want to know their total antibody concentration in that sample. I want to know what that is. So here is my complex right here. Here's my antigen, and I would bind it to the well. Then you would add your sample with the antibody. Maybe it's blood, maybe it's something else, some other type of saliva. And then I would add a labeled secondary antibody that has that enzyme in there, so that when I add a substrate, it will change shape when interacting with that enzyme and produce a color change that I can see or that a machine can read and quantify. So what are the benefits of using this method, or why you would use it? Well, like I said, it kind of goes back to whatever your question is, right? But also, this is a more flexible system, so there's higher flexibility than the direct ELISA. There's signal amplification, so that can be helpful. But because there are more steps, it's more complex. So with more steps, right, more human error, more different things that could go wrong, potentially, right? And then you have potential for cross-reactivity of the secondary antibody. So we don't want that. That would not be great. But this is something that people would use, like I said, if you were in a hospital and you were trying to like measure antibody concentrations in a serum or a sample from a patient. This is what would be used. In your lab that we're doing, what we're looking to do, and a lot of the different components of each step, so when you actually look at your procedure, you'll need to know why we use blocking buffers and why we use the different things that we do. That's part of the post-lab questions. And the answers for that can be found on the bottom of page 33. So take some time and read that. It'll really help you in answering those questions. All right, let's talk about antigen capture ELISA. So antigen capture ELISA is also sometimes called sandwich. Just so you guys know, and if you hear it somewhere else, or maybe you've heard it in the past, that is what that is. But what you're doing is you're trying to detect the presence of a certain antigen, or to detect the presence of an antibody against a particular antigen. So compared to the other ones, if you recall, we had the antigen bound to our plate or well. In this one, the antibody is bound to that well. And as you can see, the antigen here is sandwiched between these two antibodies. And our label antibody is right here. And here's our substrate, so that when it gets in contact with that enzyme, it'll change, and like I said, produce that color change that we can see. So when would you do this method? Let's say that your antigen, when you bind it to this plate, like I mentioned previously, bound to the matrix, it can change shape. Let's say that that's what occurs with your particular antigen. Well, then you might want to do a sandwich, an antigen capture ELISA, because you don't want it to change shape, because changing conformation could affect the binding of these antibodies, right? So this is something that you would do in that case. There's more flexibility for this particular assay. It's more sensitive, but what you need to have is a large enough antigen so that both of these antibodies can bind. If it's too small, there might not be enough space for it to bind on that particular antigen. And sometimes it can be difficult to find a second antibody that also binds to your antigen. So that can be some hindrance to this particular procedure. All right, let's talk about antibody capture ELISA. So what you would do in this one is to detect the presence of a certain type of antibody. So here's the image that I have associated. Let's say that you want to see if a patient is mounting an immune response against a particular antigen. You want to see if that immune response is there. This is the type of test that you would do. So here's the matrix right here, right? We note that there's also an antibody bound to it. But what you have here is after you have that bound antibody, you'll add the serum antibody, the sample from the patient, maybe blood, saliva, whatever. Then there's the antigen, right? And then there's a labeled antibody and our substrate color changed. So higher flexibility again with this, higher sensitivity again with this. But again, the antigen must be large enough to where these two antibodies can bind to it. And you still have that same issue of finding a second antibody that also binds to your antigen. So that can be an issue sometimes as well. But each of these have been developed to answer particular types of questions. You might come across these if you're working in medicine, if you're working in a research lab. It all just really depends on the type of question you're asking and what you're trying to look for. So when you go into the future and you go into the world, and if you have to do an ELISA, or if someone's talking about an ELISA, you'll know what it is, hopefully, and you'll know which one that maybe they did, maybe if they're testing on you, or if you're running a test. Awesome. Well, good luck on your post-lab, and I can't believe that this has gone so quickly. This is the last lab. So you should be excited. Pat yourself on the back, and as a reward, here is some pictures of my dog Tucker.